Good afternoon, everyone. It's always a pleasure to be able to be here on Wednesday afternoon to introduce the speaker. It's a special pleasure when the speaker happens to be a long-term friend, and that is the case today, as I have the opportunity to introduce Raju Kucherlapati, who I have known for at least 20 years, probably more like 25, as we have labored together in a very exciting field called genomics, as it's increasingly applied to lots of different human conditions, and especially, as you'll hear about today, Dr. Kucherlapati's contributions to cancer are adding to our wonders at what we can learn about this disease. His education began in India, where he got his undergraduate and master's degree in Andhra University, and then he went on to get a Ph.D. in genetics at the University of Illinois in Urbana. After postdoc with Frank Ruddle at Yale University, he then went on to several academic institutions of note, Princeton, University of Illinois, Albert Einstein College of Medicine, and most recently at Harvard, where he is the Paul Cabot Professor of Genetics, Professor of Medicine at Harvard Medical School. His research has covered many areas of human genetics. He was certainly a major contributor to the Human Genome Project. We all spent a lot of time together in those days trying to make sure the project succeeded, and happily it did so. He has done significant work, in particular human genetic disorders such as Noonan syndrome, and he's been a very significant contributor to the Cancer Genome Atlas, TCGA, a joint effort between the Cancer Institute and the Genome Institute. He has also given of himself to many other important endeavors, and I will mention two, namely that he is currently a member of the National Advisory Council for the National Human Genome Research Institute, and is also a member of the Presidential Commission for the Study of Bioethics, where he contributes important insights into many of the ethical dilemmas that are arising from our newfound abilities to read genomes and other interesting aspects of our personhood. It has been, over the course of these many years, a delight to be able to converse with Raju about many areas of science, and I'm delighted that he's here today to speak to you about the genomics of cancer. So please join me in giving a warm welcome to Dr. Raju Kucherlapati.
Francis, thank you. Thank you very much. I think I'm not going to stand behind the podium, because when I stand behind the podium, nobody can see me. It's a great pleasure for me to be here. I spent many times here at the NIH in a varieties of different ways, and of course grateful for the NIH for all of the financial support that I have received over the years. So I want to tell you today a little bit about how our knowledge about cancer is changing the way that we think about therapeutic approaches to cancer. So this is the tale of two presidents. First of all, I think that many of you probably recognize that it was during the time when President Roosevelt was in the White House that the National Cancer Institute was established. At the time that the NCI was established, it was recognized that cancer is an important disorder, but it was not very clear as to whether or not we'd really be able to do anything about it. And at the time that the President Roosevelt signed the bill, it was just to try to begin to understand about cancer. And over the years, things really have changed. And when President Nixon was in the White House, this was the declaration of the war on cancer. And whether or not the war is successful can be debated, but one thing that cannot be debated is that this bill that President Nixon signed really increased the amount of money that the National Cancer Institute has had over these years, or 40 years or so. And many of the people like me who have been receiving support benefited from this largesse. But obviously the reason why the people of the United States give us money is to really try to solve the problems and try to cure diseases. So it is important to be able to ask what has happened to this important disorder, and what kind of progress have we as a community have made. And as you could see here, I was able to find the data only not the last 5 years, but until 5 years ago. And as you could see here, that in all of these types of cancers, the overall survival rates have begun to get better, but we're not quite there. If you think about certain types of cancers like lung cancer, for example, the long-term survival rates are very good. Pancreas cancer, equally not very good. So one of the questions is that if we want to be able to increase the survival rates and indeed make cancer history, what do we need to do? And so one notion is that we need to be able to try to gain a better understanding about cancer. And so how can we think about that? And there are many, many different approaches to understand this problem. But one approach is a genetic problem. And we all know that cancer is a genetic disease. And the reason why we know that cancer is a genetic disease is that germline mutations in a number of different genes cause cancer predisposition syndromes. In many of these cases, the penetrance with which these individuals develop cancer is very, very high. The other piece of evidence is that many of the genes that are involved in this cancer predisposition, such as the adenomatous polyposis coli or APC gene that causes a hereditary predisposition for colon cancer, germline mutations in this are present in virtually all colon cancer, sporadic colon cancer genes. And over these last many years, we have identified a large number of tumor suppressor genes and oncogenes, and mutations in these genes are found in cancers. And we're beginning to recognize that it is not just mutations in genes that are important, but a varieties of other types of changes, such as copy number changes, DNA methylation changes, changes in the levels of expression of genes and proteins are also very, very important. So the notion was that would it be possible to be able to try to develop a comprehensive understanding of all of these genetic changes, and if we do gain such an understanding, that that it would help think about therapeutic approaches. And so this endeavor now really became possible from the Human Genome Project. And as Francis mentioned, I had the privilege of being a part of the Human Genome Project, and all of you recognize that the Human Genome Project not only provided us the blueprint of the human genome, but also developed varieties of new technologies that came alongside with that, and all of these technology advancements and the relatively lower expenses of being able to sequence the genomes is really making it possible to be able to undertake a comprehensive understanding about the genetics and genomics of cancer. So to accomplish this goal, the National Human Genome Research Institute and the National Cancer Institute established this program called the Cancer Genome Atlas, and the particular name is attributed to be given to this program by Francis. And so the TCGA stands for the Cancer Genome Atlas, and the goal of this program is to examine large numbers of solid tumors. In each of these cases, to get 500 tumors from patients who've been untreated, and to obtain nucleic acids from them, and also obtain blood samples from those same individuals, and conduct a variety of different types of analyses for all of these. And one of the wonderful things is that you would be able to do, apply a varieties of different types of genomic methodologies with the same samples. So therefore, this is an unprecedented program to be able to, try to gain such an understanding. And to accomplish this, a number of different types of centers that have been established, and one of those kinds of centers are the so-called cancer genomic characterization centers, and I have the privilege of running one of them at Harvard. And so I won't tell you about all of the different things that the TCGA has been doing and I'll take a little bit of mostly the work from our group, but a little vignettes from other sorts of places to tell you, how things are changing. And I want to emphasize today as to how this information is actually helping us to think about therapeutic approaches for cancer. So one of the approaches for this is to be able to do sequencing of DNA. And there are varieties of different types of sequencing that you could do. One is to be able to look at whole genome sequencing. This is what we do in my group. It is also possible to be able to sequence the exomes from the tumors and the normals. And it is also possible now to be able to sequence all of the transcriptome from each of these different sets of genes. And so let's take some of these things and how do they work. The first one is to be able to try to use this information to determine copy number changes. And this is done in a very simple fashion that is illustrated here on the upper left-hand side. And the way that it is done is you sequence the tumor DNA and the normal DNA and segment the genome into small bite-sized pieces and just count the number of sequence tags that are present here. So when you see the cases in the controls, if approximate number of reads that are present in the tumors and normals is the same, that means that that region has not undergone any types of copy number changes. If the number of reads that are present in the tumor increase relative to the normal, that means that there is an increase in copy number, and the reverse of that is that if there would be copy loss, you'd be able to detect that. And the picture on the bottom, really shows, for one tumor, this is a colon tumor, as to what the profile for the entire genome is. And as you can see, there are large numbers of the genome that are unchanged, and there are some segments of the genome where there is increase in copy number, and there are others that are reduction in copy number. So you take one tumor, that's what it looks like, and here are the data from 500 tumors and what they look like. And in this slide, all of the red and blue that you would be able to see, those represent regions of amplification or loss of a copy of the gene. And I just want to point out that basically that there are a whole bunch of tumors that are seen up at the top of the slide, which are sort of light. They don't have any color, either blue or red. And actually it turns out that these are a bunch of tumors that do not have much copy number changes. And what is also really very interesting about this is on the right-hand side, it actually shows the number of somatic mutations that are present in each of these tumors. And so what is really interesting is that all of these tumors that have these virtually stable copy number changes have a very high level of mutations. So where do these high mutations come from, and do they have any clinical significance? So you can ask that question, and I'll illustrate this point by looking at data from colon cancer. In this particular case, the number of mutations that are present in 224 tumors are plotted. And as you could see, that there are some tumors that are at the extreme right-hand side that have as few mutations as 1 per megabase of DNA that is sequenced. And there is a continuous range of them. And on the left-hand side, you'd be able to see that there are some tumors that have as many as 800 mutations per megabase. And these can be classified into these two categories, or that we actually classified them in three categories. All of the tumors, there are a majority of them which are on the right-hand side, which have types of mutation frequencies that are comparable to many other tumors that are seen. But a group of tumors on the left-hand side shows their high levels of mutation, and those few tumors that have very high levels, like more than 100 mutations, are classified as ultra mutated tumors. So why would a genome contain very high numbers of mutations, and what are the possible types of mechanisms that would result in those types of changes? And there are basically one mechanism that is well understood as to how tumors or cells would acquire high levels of mutations, and that is shown on the right-hand side. And as all of you may know, that every cell, mammalian cells or other organisms, have a system to repair mismatches of DNA that might occur either due to errors in DNA replication or other types of changes. And there is a system called the DNA mismatch repair system that is capable of recognizing these types of changes, and they're capable of repairing those mutations. And it turns out that germline mutations in these genes can occur, and those germline mutations actually cause cancer predisposition syndromes. And somatic mutations in these genes would also result in high levels of mutation frequencies. A second possible way in which mutations can occur is actually through errors caused by polymerase itself. As you know, there are two polymerases that are important in DNA replication. One polymerase that is responsible for leading strand synthesis, and another polymerase that is responsible for lagging strand synthesis of DNA. And these polymerases have two domains. One is a catalytic domain that is capable of introducing the right nucleotide, and the second domain is a proofreading domain that recognizes any errors in the polymerase function and is capable of repairing them. So one could imagine that mutations in this polymerase might also cause an increase in mutation rates. But such mutations have not been seen before this project has started. So we looked at all of these sets of different genes, and the results of that are shown in this inset in the upper right-hand corner. And basically what it shows is that now those tumors that have these hypermutated or ultramutated phenotype, if you look at them, each of these tumors have either mutations in one or more of the DNA mismatch repair genes, or interestingly, these ultramutated tumors that have very high levels of mutations have polymerase E, the leading strand DNA synthesis enzyme. Most interestingly, all of these mutations that are present in POLE are present in the proofreading domain, and no mutations are found in the catalytic domain. Of course, it would make sense, because if you have a mutation in the catalytic domain, the enzyme would be dead, and that wouldn't be good. And of course, if you have a mutation in the proofreading domain, the enzyme is still functional, and that will result in this phenotype. So what does this got to do with the therapy? And before I tell you about the therapy, the question is whether this type of phenomenon that we talked about is restricted to colon cancer, or whether this is true in other types of cancers. And it turns out that this is not restricted to colon cancer, and these are results from endometrial cancer, and the same type of phenomenon also occurs in endometrial cancer. There are a bunch of tumors that are polymerase epsilon mutations. Some of them have these mismatch repair mutations and so on. But from a clinical point of view, an interesting observation is that it turns out that in this particular case, those patients whose tumors have these polymerase epsilon mutations, their long-term survival is excellent. And you can see this over several years that these patients essentially, none of them die, whereas those other patients with other types of changes don't have as good a survival. Obviously, this is very important from a clinical point of view, that so if you know which patients are the ones that would have a better outcomes as opposed to which patients are not going to have better outcomes, the approach that you would use to be able to treat those patients is going to be completely different. Right? And the other feature that is clinically important is that because of these differences in the survival, it is sort of suggested that patients to be tested for mutations in DNA mismatch repair systems. And what these results show is that it's not sufficient to just look at those genes that are involved in DNA mismatch repair, but we also be needing to look at the polymerase E or epsilon mutations. So we need to think about a completely different way of analyzing these tumors and the mutation frequencies to be able to make clinical decisions about how we would be able to treat with them. The second feature that's actually quite interesting is that when you look at these changes that are present in them, they can be represented in these so-called circos diagrams. And the outside of the circle diagrams show each of the chromosomes. And inside, in the blue, it shows the intrachromosomal rearrangements that you see. And the red lines show interchromosomal recombination events. And so what's really interesting is that certain sets of tumors that you'll be able to see in the first tumor on 3685 that's on the left-hand side, there is a region, for example, that contains a tremendous number of rearrangements in a small portion of the genome. And that is true, as you could see, in all of these 5 different tumors. This phenomenon has been described many years ago, and it is called the chromothripsis. And if you look at it very closely, this is what happens. If you look at a small portion of the genome, like on chromosome 6 at the top, and chromosome 1 in the middle, and chromosome 12 in the bottom, portions of the genome shows copy number changes, which are usually either changes from 2 to 3 or 4 copy number changes, but all of those orange lines represent the tremendous amount of rearrangement that occurs. So what is happening in these particular cases is that the region of this chromosome gets broken down, undergoes double-strand breaks, and all of these small fragments now randomly rearrange to give rise to the changes in the copy number changes, as well as these tremendous amounts of rearrangements. So what is the biological significance of such events that are present in a subset of the tumors? There are a couple of things that are important. One is that if there is a gene that is like a tumor suppressor gene that is within this region of chromothripsis, obviously this type of event is going to result in inactivation of such a gene. Another type of event that occurs is that there are certain oncogenes or other types of genes that are important for progressions of cancer, initiation of cancer, that these rearrangements might be able to bring in a new promoter or new types of rearranged genes. And there are indeed examples in each of these cases of specific genes that would be involved in that process. So it turns out that this process of chromothripsis was not appreciated about the extent to which it occurs in tumors, but it actually turns out this is a very virtually all solid tumors have these types of chromothripsis events, and the frequency with which these events occur is not very high, but this is yet another mechanism of either turning off or turning on genes. So you can use this type of information not only to look at these types of changes, but you can also look at genes that are amplified. And again, here is a set of results from the analysis about 225 colon tumors, and the left-hand side shows all of the regions of the genome that show amplification in a significant fashion. I'll just illustrate, with one of them, what the interesting and important feature is. One of those regions is on chromosome 11, and so that is shown here. And in this region that is amplified in these large numbers of tumors that are shown by these red bars, there turns out to be 4 different genes. One is IGF-2, insulin-like growth factor 2, and within that gene, in the intron of one of them, is a microRNA called MERV483. There is a gene called tyrosine hydroxylase that is important in melanogenesis, the insulin gene itself, and there is a gene called ASCL2, which is involved in stem cell biogenesis. And all of these genes have been considered to be important, in colon cancer, and but we had the opportunity to actually examine whether all of these genes are important or only one of these genes is important in colon cancer. And so fortunately, by virtue of the fact that we were able to have RNA data, expression profile data from all of these tumors, we were able to examine and correlate the amplifications of this region with RNA, and that is shown in the bottom two slides. And what we show is of all of the 4 genes that are present, the only 2 genes whose levels of expression are altered are IGF2 and MER483. So that would clearly tell us, number one, that of all of the 4 genes that have been involved or implicated, only one of them, the IGF2 and the MER483, which is encoded within that, are the ones that are critical and important. So if you look at all of these, it turns out the total number of tumors that have this IGF2 overexpression, constitute about 22% of the tumors. And IGF2 is in the PI3 kinase pathway. So one could ask the question then, are the IGF2 mutations important in cancer? And one way that you would assess is that, whether the mutations or alterations in this gene are mutually exclusive of other types of changes in other members of the PI3 kinase pathway. And this result in this slide shows that indeed all of those tumors that contain IGF2 or MER483 alterations are mutually exclusive from insulin like receptor or the PI3 kinase catalytic domains or loss of PTEN. It would suggest that actually that this is important. And so if you add up all of these different sets of genes that are modified in this pathway, it turns out that 54% of the tumors have changes in this PI3 kinase pathway. From a clinical point of view, this is very important because it turns out that there are currently drugs in development that would that are targeted to all of these different things. There are IGF receptor inhibitors, antibodies, drugs that are capable of inhibiting the PI3 kinase pathway that are currently in development in other diseases. And that means that all of these colon cancer patients can enter into clinical trials with these drugs. And I'll tell you about where that effort is in a short while. Another example of this amplification that really turns out to be very interesting is that in about 4% of the tumors that were examined, there is an amplification of ERB2. ERB2 amplifications are well known in breast cancer, and there is a drug that was approved in 1998 called trastuzumab, and women whose breast cancers have amplification of ERB2 are treated with this drug, and they show excellent response with this drug. And when this discovery was made in the case of colon cancer, that is to suggest that because it is also amplification and overexpression of ERB2, that these patients might also benefit from treatment with the trastuzumab. And what is also interesting is that this ERB2 amplification, originally described in breast cancer, is now found to be present in small proportions of not only colon cancer, but also in lung cancers, and therefore that both of those cancer types, that those subtypes can indeed be, tried in a clinical trial with this drug. Now, so using these types of approaches, it is possible to actually look at, what are the types of pathways that are actually affected. And one of those pathways that is known, to be involved in colon cancer is the Wnt signaling pathway. And the canonical mutations in the Wnt signaling pathway that were known to be present in colon cancer are the gene that I mentioned to you, APC, and a large proportion of the tumors have APC mutations. So we were able to go and look at not only the APC gene, but many members of the Wnt signaling family. And all of the genes that are shown here on the left-hand side in the Wnt signaling pathway are found to be altered in colon cancer. So what this actually tells us is that, it is not just one gene that is important, but there are many different ways by which this pathway can be altered, and the cancer cells use all of those, mechanisms, to change the pathway. And these results that I showed you here about the genes that are actually involved in them are copy number changes and mutations that are present. It also turns out that there is another very interesting mechanism by which this pathway is altered, and that is shown here. This whole genome sequencing, when you look at them, it turns out that the way that it's done is that a piece of DNA, usually 300 base pairs long, is sequenced from both ends, and both ends that are sequenced with 50 nucleotides or so are mapped back to the genome. So you'd expect that both of those ends of those sequence fragments would map about 3 kb apart. Occasionally, however, they do not. Here's an example of one of those. And when you sequence them, all of those gray regions are sequences, and the red dots really represent these so-called split reads. And this particular fragment, one maps to this gene TCF7L2, and the other end of the fragment maps to this gene called VT1A. So what is the effect of this? And in this particular case, it turns out that this rearrangement that involves these two genes is rather complicated, and but the ultimate goal of what happens all through this rearrangement is shown at the bottom. That the red here is all of the transcripts are shown here. The red shows increased level of expression. And basically this rearrangement now resulted in an overexpression of this gene called TCF7L2. But what is TCF7L2? TCF7L2 encodes a gene that is a transcription factor that is present in the nucleus. Under normal circumstances, it is a transcriptional repressor. But when the Wnt signaling pathway is turned on and beta-catenin, one of the regulatory molecules, moves into the nucleus, it's bound to the TCF7L2, and that results in the transcriptional activation of many oncogenic and growth regulatory pathways, including MYC. And so this is a completely different way in which this Wnt signaling pathway gene is altered by rearrangement. And so the question is, if you find such rearrangements, are they important, or do they have biological significance? And my colleagues, Matt Meyerson and Adam Bass at Harvard, have done the experiment of taking one of these cell lines that contains such a translocation and they use shRNA to inhibit that, and it actually turns out that if you do that, then you kill the cells, clearly showing that this is a driver gene. So these particular ones are present with in different studies with different frequencies, but about 8% of the colon tumors have these types of changes. And so that is just actually a small subset. And here is a whole bunch of other types of genes, translocations that involve members of the Wnt signaling pathway, all of them in colon cancer. And so we don't have to look at each one of them separately, but you could see that there are a large number of genes that are turned off or turned on that will result in an increase in the pathway. So now you ask the question, what is the importance of these? And this is also, each of the tumors is on the vertical axis, and be able to see all the different types of changes that are present. And you could see that most of the tumors that have been examined have mutations in APC, well established, but a whole bunch of other types of changes are present in these. And what this result would suggest to us, the fact that some of these types of translocations are shown to be functionally important, is that more than one way in which you would be able to alter this Wnt signaling pathway has a selective advantage in these particular sets of tumors. And the other thing that it tells us is that when you're now thinking about, let us say, developing Wnt signaling inhibitors, it is not sufficient to just look at the APC gene or the beta-catenin gene and its genetic status, but you need to look at all of these different sets of genes and understand their status. Otherwise, you would be able to leave some critical patients out of our kinds of studies. And so this is So this also would suggest that the old way of trying to look for mutations in one genes or two genes is not going to be sufficient, and you need to be able to use the modern approaches to try to analyze the genes and genomes of these tumors before you make clinical decisions. Is this true just for a Wnt signaling pathway? It turns out that this is true for all of the pathways that are involved and implicated in cancer. And one other pathways that we all know very well are the PI3 kinase signaling pathway and the RAS signaling pathway. And like mutations in KRAS and NRAS and BRAF are now present in many, many different types of tumor types. And again, we show just here in this picture, just genetic changes in these tumors. But what I want to show you in this next slide is that it turns out that if you look at these pathways, many, many of these genes are now also subject to rearrangements, like the ones that are shown, like in the case of RAS genes. All of those things that are shown in the red, for example, they're all involved in chromosomal translocations, and in many cases that you can actually directly show that those such translocations indeed result in changes in expression of the gene, suggesting that they have functional significance. The other feature that's also very interesting and suggests that these would have functional significance is this mutual exclusivity analysis that is shown at the bottom. At the top of these two lines that you show all of the tumors that have either HK or NRAS mutations, and the bottom line shows all of the tumors that have these kinds of structural aberrations. And you can see in all of those cases that have these types of structural aberrations that are shown on the right-hand side, that they do not have point mutations. So that again suggests that there's an alternate way in which you would be able to change the expression and the patterns of these tumors. There are other types of things that you'd be able to see, and here are some results from lung cancer. These structural aberrations that are found, and the interesting features about them is that when you do this type of analysis, certain types of structural rearrangements, such as EML-ALK4 that is well known, for which there is a drug that's available right now, is detected. But there are a whole bunch of other types of things that are related, tyrosine kinases that are involved in these structural aberrations, and all of them result in activation of these genes. Again, it suggests that for those cases where there is, because these are kinases and they're susceptible for drug development, that there are opportunities for new drug development, using these types of targets. Now, how could all this information be used in thinking about therapeutic approaches? And this slide actually shows emphasizes the point that I was trying to make earlier about understanding these pathways. So what we did for all of these different tumors that have been analyzed, we looked and plotted which of the pathways are actually altered. And again, each vertical bar represents one tumor. And so as you could see, there are certain types of tumors in this particular case that have the Wnt signaling pathway, the p53 mutations, RAS, activating mutations, PI3 kinase mutations, or TGF-beta, loss of function mutations present. And you could see a whole spectrum of different types of mutations that are present. So the importance of this is that if you're actually thinking about therapeutic approaches for these patients, you need to know this information. For example, it has been known in animal models that if you take a mouse that has an activation in the RAS MAP kinase pathway and try to use a MEK inhibitor, which is a downstream activator molecule, it turns out that those mice would develop transient response, but they become resistant. And when you look at them, it turns out all of them have now turned on the PI3 kinase pathway. And that So one needs to know then that what that means is that if you take the RAS mutations in these patients and then try to give them MEK inhibitors, they're not going to work. You need to understand that some of these patients, you need to inhibit both RAS-MAP kinase pathway as well as the PI3 kinase pathway to be able to have an effect. And that's obviously important. So this kind of information is interesting. But the other interesting piece of information about this is that this type of feature of overlapping sets of genes that are mutated is not common to all cancers. This is somewhat unique to colon cancer. In lung cancer, it turns out that much of the mutations are mutually exclusive and the pathways are unique, as I'll show you in a second. But in this particular case, it's important. So what it means is that we need to be able to look at all of the genes that are involved in each of these pathways to understand whether they're turned on or turned off, those pathways, before you could think of an appropriate therapeutic approach for them. Now, I want to just give one more illustration about the importance of these structural aberrations, and that comes from the study with the melanoma. And again, we looked at large numbers of melanoma patients, and as you would expect, it turns out that a significant number of them have BRAF mutations, a bunch of them have NRAS mutations, and so on and so forth, that are shown by these red regions. But there are a bunch of tumors up at the right-hand corner that do not have any of these types of changes. What could be the reason? Why? What is the driver for those? And it turns out that if you look at these, all of those tumors at the right-hand end indeed contain some type of structural aberration that are shown here. And all of those structural aberrations that are shown down the bottom, they're an important genes. MDM2, the regulator of p53, AKT, which is an important component to the PI3 kinase pathway, RAB genes, which are important in MAP kinase pathway. These structural aberrations in these tumors, indeed turn on all of these different sets of genes, suggesting that those tumors have turned these functions on, and we know only about one of these right now in a functional assay indeed turns out to be an activating change, whereas it's most likely that the other ones are too. I would suggest that these types of structural changes, that has been recognized to be important in hematologic malignancies, are not as well appreciated in solid tumors, but they also play a very, very important role. All right. So one other feature that's actually important from these studies, using these types of genome sequencing efforts is that, the way that the analysis is done is that all of the DNA sequences obtained is then mapped back to the human genome, and anything that doesn't map to the human genome is left aside. And it actually turns out that the DNA that is left aside that doesn't align to the human genome has an enormous amount of tremendously valuable information. And one of them is mitochondrial DNA. And even at very low coverage of the genome, you have a huge coverage of mitochondrial DNA because of the number of mitochondria that are present and the small amount of genome that is present. And so one of the really very interesting things about the analysis of mitochondrial DNA, I'll show you a couple of examples, is that all of you know that the energetics of the cell normally uses the oxidative phosphorylation system from the mitochondria, and the other energetic system is that if this fails, is to use the TCA cycle. And that is also known as the Warburg effect. And it turns out that in a large numbers of these tumors that you see, it actually turns out that one or more of the genes in the mitochondria suffer mutations. And also most interesting, it turns out that these mitochondria initially start out as heteroplasmic, but in the tumors, those that contain these mutations become homoplasmic. This is suggesting that there is a strong selection, for them, and this is true in a bunch of cancers that have been examined, and other types of genes are also, genes that are known to be involved in causing childhood disorders such as MELAS syndrome. Those germline mutations cause their mitochondrial disorder called MELAS, but actually somatic mutations of this mitochondria are found in these tumors. It is also clinically significant because there are, it is possible to be able to sort of develop synthetic lethals if the cell is now completely dependent on using the TCA cycle. Inhibition of that pathway would essentially, abrogate the energetic functions of the cell, and you can kill them. And there are several such drugs that are currently in development. So given all of this piece of information, now what are the possibilities for colon cancer? To understand about the therapeutic approach for colon cancer, for the longest period of time, and even today, there are two chemotherapeutic regimens that are most popular, FOLFOX and FOLFIRI. It is a cocktail of chemotherapeutic drugs that are used, and virtually every patient gets them. There is EGF receptor inhibitors that are used in these patients, and an angiogenesis inhibitor is also approved for this indication. Now, when we look at all of these different types of changes that we find, indeed it turns out that there are currently drugs in development for Wnt signaling pathway, and 95% of the tumors have changes in Wnt signaling pathway. RAS mutations, 40% of the tumors have RAS pathway mutations. BRAF mutations, 15% of these tumors have BRAF, and we have approaches to think about them. IGF-2 that I told you, and the PI3 kinase, and together they constitute about 50% of the tumors, ERB2 amplification, and so on and so forth. So now, a cancer type for which there wasn't really much of a therapeutic opportunities and a very little targeted therapies, now we have the opportunity to be able to think about, a large, trial. And there is currently the National Clinical Trials Network is contemplating and planning on doing a clinical trial in which it plans to recruit about 3,000 patients and examine them for all of these different sets of genes, and then assign individual patients to specific therapeutic approaches based upon the genetics. And as you know, that there is a similar trial here at the NIH called the MATCH trial for all types of cancers and being able to do these types of analysis. And there is also another trial for lung squamous cell carcinoma that's also doing these types of analysis. So in a few years that we would be able to have information about how to deal with them. So I just want to close by saying that what I told you primarily focused on colon cancer, but this is not restricted to colon cancer. Lung cancer, this is an example. Now 11 different types of genetic changes are known, and you can classify the lung tumors based upon this. And every one of these subcategories of lung tumor patients, there is either an approved drug or a drug that is currently in development. And so at our institution, all of these patients, lung cancer patients, are tested for all of these different changes, and the nature of the treatment that is provided to those patients is dependent upon the genetic profile of the tumors. And it's a paper that is published by my colleague Levi Garraway, and Levi shows that that it is just not the lung adeno or lung squamous or breast. All of these different types of cancers can be classified into these subcategories or stratify these patients based upon these genetic differences. And interestingly, it turns out that all of these types of genetic changes that are seen, they have either drugs that are approved or currently in development. So we can anticipate that in the next few years that as some of these drugs get approved, that we'll be able to increase our capacity to be able to do what we want to do. So to summarize, I think our knowledge about cancer as a result of this large-scale effort is increasing. and the knowledge is increasing at a very rapid pace. And we're beginning to understand the role of many, many different types of genetic and genomic changes that play a very important role. And we're also beginning to better understand the different ways by which the different biochemical pathways can be altered, and most importantly, from the point of view of the patients, that many of these changes are associated with either approved or emerging therapies. And I want to close by acknowledging all of the members of the TCGA, and many of them are involved, and my own colleagues at Harvard. And the program was started in association with Linda Chin when she was at Harvard. She now moved to MD Anderson, and all of our colleagues who contributed to this effort. Thank you very much.
Well, terrific. We have time for some questions. Please, if you have a question, come to the microphone where people can hear, including those who are watching by video. And we seem to have a question already. Yes, please.
Thank you. I have a fundamental concern about using site-specific cancers and identifying mutations in the middle of a very chaotic disorder, the structure or unstructure, and identifying too many different genes which are mutated, and of course they should be mutated when they are growing two cancerous cells. Why aren't we focusing on the right focus, which is to see what causes the tumoricidal versus tumorigenic pathways of immune or cancer surveillance, which has been known 100 years ago, and we accidentally bump into it when we were in the inflammatory diseases. Why aren't we looking systematically to see what causes the immune dysfunction that we come to cancerous cells? Otherwise, there is dysfunction in autophagy, mitophagy, and so forth and so on. Many decoy receptors are there. And of course, you would expect that the changes in the balance between wound healing, which are growth factors, and apoptosis should change in the middle of cancerous cells.
Raju.
That's a great question. Thank you. So the answer is yes, there are efforts that are underway. My talk, just deals with the genetic and genomic changes. But there are actually, as you may know, there are some very exciting developments in understanding the immune system and the role that it plays in tumor surveillance and so on. And there are a number of genes that are known to be involved in immune checkpoints that have now been identified. And some of those, like the CTLA4, which is a very well-known molecule, and antibodies against CTLA4 have been approved for therapy in case of melanoma, and they're actually showing fantastic results. And there are many other molecules like PD-1 and PDL, and there are many of them are in clinical trials currently. There are also immune checkpoints, and those are also showing really extremely promising results. So I think many people think about the way to approach them is not to think about just one approach. Identify the genetic and genomic changes, try to stratify the patients based upon that, and try to get a drug that will be able to inhibit the pathway and get the immune system to work at the same time. And using both of those approaches might ultimately turn out to be the very best way in which we'd be able to care for cancer patients.
Great. Thanks. Hey, Raju, you talked about this interesting phenomenon of chromothripsis, where a chromosome just gets shattered into all these little pieces, and yet you showed those circus plots which showed that in those same tumors, most of the chromosomes aren't affected. It seems to be a specific chromosome that just gets blasted to smithereens. And it's hard to kind of imagine what's happening mechanistically there that happens on a chromosomal level, not even a regional level, but the whole chromosome seems to be affected by this. Do you have an idea mechanistically what happens there? Is this something that happens in one step, or is it an original rearrangement that sort of destabilizes the chromosome and then actually over many different cell divisions ends up in a scramble? What do we know about that?
So, Francis, this has been considered to be a catastrophic event that occurs once. And the mechanism by which it occurs is proposed to be the following. As we know that under normal circumstances, there is normal disjunction of homologous chromosomes and mitosis, but occasionally some chromosomes lag behind and they enter into these micronuclei. And so now what happens is that then each of the nuclei would start replication processes, and so this one chromosome that is sitting in one micronucleus, its DNA synthesis is supposed to be completely discombobulated from the rest of them, and that is proposed as a mechanism. But we don't have direct evidence to support that, except from these kinds of studies show that clearly what is happening is that region of the chromosome undergoes significant amount of double-strand breakage.
But you haven't caught one in the act.
That's correct.
Over here.
Have you found any distinct associations in correlating with your studies with histone acetylation or methylation? In other words, has epigenetics entered into your equation? Because it sounds like it would explain, you're saying everything's blasted to smithereens, so there's got to be something controlling the chromosomes in the first place. And now, as you know, epigenetics is a big deal, and the histones are a big deal, and acetylation and methylation would seem to play into what you're talking about, no?
Okay, good question. First, globally, there is significant amount of role that DNA methylation plays in all of these tumors in a varieties of different ways. Like, for example, this hypermutation that I talked about, many of those cases that actually result from the methylation and the methylation-based inactivation of MLH1 gene. So that's very common. It also turns out in many of these tumor types, many of the genes that are involved in the methylation and maintenance of methylation are actually mutated. So DNA methylation plays a very important role. In the case of chromothripsis, what the role of DNA methylation plays is not known.
One other question. You were talking about cancers per se, but you weren't talking about very well-known and important precancerous states. For example, in endometrial cancer, you'd have endometrial hyperplasia, which is a diagnostic dilemma for most pathologists, in which this type of genetic analysis would be very helpful. You would have dysplastic polyps in terms of colon cancer. That would be very helpful if there was a good marker like that. You have atypical intraductal hyperplasia in breast lesions for pathologists, and nobody can tell which ones are going to become cancer or not. It's just a big question mark. And in terms of lung cancer, you'll have bronchial dysplasia, epithelial dysplasia. Those seem to be like meaningful clinical questions that could be addressed via this in terms of diagnostic testing. That's all.
You're absolutely right. So first of all, with regard to TCGA, one of the constraints was that we had to get tumors from patients who are untreated, and the tumor should be a primary tumor, and it should be an excised tumor, and it should provide enough nucleic acids to be distributed to all of these different centers to do all of these different types of analyses. So we were constrained by that. And many of these other types of things that you talked about, they're tremendously interesting to look at and to be able to see when does these changes occur and whether interventions at early stages are important or not important, or what are the kinds of interventions that we could have. And so one of the things some of us in the scientific community have been proposing is to not look at the 10,000 tumors that we look at, that we should have a program to look at 100,000 or more tumors of all stages and all types. And as you know, all of them are constrained by the amount of money that we have.
Yeah, there is that. Well, on that note, I'd like to certainly invite all of you to a reception in the medical library with Dr. Kucherlapati where you can continue the conversation. But please, let's thank our speaker once again for a wonderful presentation.